Good afternoon, everybody. I'm going to introduce you to Immunicum. We are a publicly listed company, NASDAQ Stockholm. So I need to show you also the disclaimer. So what is Immunicum? Immunicum is a company operating in the immuno-oncology space, as several other companies already presented today. But we have a differentiated product. We are a cell-based therapy with key attributes. It's allogeneic and off-the-shelf. And we are already in phase 2. As a matter of fact, we completed the phase 2 recently, and I will show you data on that. We also have excellent safety profile, what of course, as you know, is very important in the treatment of cancer that is evolving to the use of combinations. The main side effect profile is fever because we induce an inflammatory response, grade 1, 2, treated with paracetamol. We are currently manufacturing with BioNTech in Germany, but transferring the process to HCAT Hitachi in the US as they have commercial scale capabilities. We have been interacting with the authorities, what, as you know, for cell-based therapies is very important already for several years. We have an IND in the US since 2016. We received the last year TMP certification from the European authorities. So it's a very good scientific validation. But also, a year ago, we entered into a collaboration with Pfizer and Merck Serono for the supply of avelumab for the phase 2 part of a new study that we have initiated. So also, as you know, there are very few of these collaborations, so also a very good scientific validation. A year ago, we raised approximately $35 million, and with that capital raise, we brought also very big institutional investors. That was very important for the company. We have now cash and runway taking us towards the end of 2021 across several value inflection points. And we have a very experienced team in the industry. So let me tell you first how we differentiate from other cell-based therapies. So our cells originate from healthy donors. These are blood donors, healthy individuals. And through leukapheresis, we extract monocytes that we convert into immature dendritic cells. We don't do any genetic manipulation, any expansion. It's purely we extract about 1 billion of these cells, and then we just activate these cells with a cocktail of cytokines to make them extremely pro-inflammatory. And when these cells start producing chemokines and cytokines, we freeze them. So it's a cryopreserved product. So as you can see, I think there's no laser here. No. From one healthy donor, we treat up to 50 patients. So from a cost of goods perspective, there's no impact on margins. But the very good thing is that we already passed the 3-year shelf life, so we really can produce whenever we want and store. And the product is also basically a universal product. You don't need to do any patient matching, and we can use the same product regardless of the patient or the type of tumor. So basically, the goal here is to activate the patient immune system. How do we do that? So when needed, just go to the hospital pharmacy, bring a small vial, is a 1 milliliter. You thaw the cells, and then you inject intratumorally. You can inject the main tumor, the metastasis, it doesn't matter. So this also gives full flexibility. And when you inject these donor dendritic cells, they are basically going to start producing these chemokines and cytokines. Massive recruitment of the patient NK cells, killing some of the tumor cells, release the neoantigens. The bystander disease from the patient are also recruited to the site of the injection, pick up on the neoantigens, migrate to the lymph nodes, and educate the CD8 plus T cells that are becoming then tumor-specific. These CD8 plus T cells then basically enter into the circulation, spread through the whole body, and all the work is really going to be done by the patient's own immune system, immune cells. The donor cells are eliminated in 2 to 3 days. But of course, in addition to the chemokines and cytokines, there are also allo cells, so they amplify this inflammatory response. But then all the work is done by the patient's own immune cells, one of the reasons why we have such excellent safety. Full flexibility, and really very wide uses of the product. As I mentioned, we can inject primary tumor, and this is no special technique. If you can do biopsy, you can inject the product, so you don't need any special equipment or expertise. So let me tell you about clinical. We have now 4 studies in the clinic. The first one was already ended in 2013. And we still have patients alive passing the 5-year survival mark. Was in newly diagnosed metastatic renal cell carcinoma patients. The data is published in 2017 if you want to get full details. Again, excellent safety. We also published in January this year a study in liver cancer. As you know, terrible disease. These patients progress very rapidly. There's not a lot of treatment alternatives. So also very, very good data there that we would like to move to the next phase. In June, we released top-line results of a study, some very small study in GIST, gastrointestinal stromal tumor, orphan disease. Patients already progressing in 3rd line, and when they were injected with our cells, stopped progressing. And of course, the one where I will spend more time, our phase 2 controlled study in combination with sunitinib, 88 patients, US and Europe. So the first thing is that these patients were all metastatic at diagnosis, so newly diagnosed metastatic. The first results that we got, that was very impressive also for some of the investigators that were in the room, was complete responses. So basically, 11% of the patients had complete responses. And according to these specialists, this is, you don't see these type of responses in these very sick patients. And so this is, when you compare and you look at other studies, sunitinib by itself normally is, 1.5% to 2% of complete responses in different studies. So really a very, very, very strong data in terms of the complete responses. But that is not only, of course, you need to see, are these responses maintained? And this is where you start really seeing even further differentiations when you compare with sunitinib alone. As you see, we had a longer duration of response. The follow-up period was 18 months. At the end of the 18 months, we also looked at all the responders, complete and partial responses, and how many of them maintained this response. You see 60% in the combination group versus 33%. In July, when we look at the survival for all patients, we also assess that 85% of the responders in our group were still alive versus 58% in the sunitinib-only group. The other thing is that all the 5 complete responses in our group were still alive in July, and the complete response in the sunitinib group had died. So very strong and long duration of responses. When we look at survival, you see here basically the 2 groups, was a 2-to-1 randomization, and you see that because of course the study was enrolling in different periods, the extended survival, and as of July, when we have the 18 months for all patients, you see already the big difference in terms of survival. Only 4 patients in our group had died, and now we are going to have in January the 2-year survival for all patients, where we expect to see further separation between the groups. Now, this strong efficacy, easy in terms of immunization for these patients are linked with a very important factor that is the safety. Basically, there was no difference in the safety between the groups, meaning that we didn't bring any added toxicity to the combination. And as you know, most of these combinations, particularly the ones that were approved recently, 71% of the patients, for instance, as an example, have grades 3 and 4 side effects. 40% of the patients cannot even tolerate the treatment. So the fact that we don't bring any added toxicity to the treatment is a very important point. And we observed this also in several of the preclinical studies where we had combinations with checkpoint inhibitors, immune enhancers, different classes, including CTLA-4. And we maintain the safety. So now we are going to have in January, again, as I say, 24 months survival follow-up for all patients. Some of them already passed 4 years. but for all patients where we expect really to see further separation in survival between the groups, including sunitinib getting to the median overall survival while we continue. And then every 6 months, we continue following this survival. Now, in addition, we are planning now the activities to meet with both European and the FDA authorities, present the data, but also discuss what will be the potential study for pivotal phase for the next phase of development. We, of course, looking at the landscape, checkpoint inhibitors are becoming, more standard of care, but with very poor efficacy in the majority of the indications. So we really enrolled the first patient in February of a study looking at head and neck cancer, non-small cell lung cancer, gastric adenocarcinoma. Very big indications, very poorly served by the checkpoint inhibitors. In head and neck, checkpoint inhibitors have about 18% efficacy, what is really, really very poor. So the phase 1b, 21 patients combining with Pembro. We already passed the first phase of the staggered, so we need to do that. And then when we move to the phase 2, we're basically going to have 3 studies running in parallel for each indication. In head and neck and gastric, we are going to combine with avelumab, Bavencio, versus Bavencio alone. And in lung, we will continue with Keytruda. So you see here, this is the protocol design for the phase 1b. We passed the first 3 staggered patients. We are now in the second phase, and then we expect second quarter next year to then move to the non-staggered phase, and then accelerating the recruitment of these patients. And basically, there was no safety concerns for the dose escalation committee when they said, yes, you can move to the normal dose. As I mentioned to you, we raised approximately $36 million a year ago. At the end of Q3, we have $34 million in cash, as I mentioned, taking us towards the end of 2021. With that capital raise, we brought very big institutional investors, including these gigantic pension funds in Sweden. And we have continued also been presenting in the US to a lot of more family offices, and we start really diversifying the nationality of our investors. So very important there. So in terms of the company, and in summary, we are in a very innovative area. We have patents granted. We are the only ones doing what we do. In addition to the IP protection, we also have the fact that we are an ATMP product that grants even stronger protection than IP. We continue developing and very good results in the clinic, showing really that we work and we make an impact in patients' lives. We have collaborations in place, raised a very good runway and attracting very strong investors. We have been doing extensive preclinical work, expanding the potentials of ilixadencel. We really want to make it as being a kind of a backbone. We can combine with different classes of drugs, kinase inhibitors, checkpoint inhibitors, even immune enhancers, and now in 6 different types of cancer and potential to be used in different ones. But of course, in addition to the efficacy, the fact that we have an excellent safety is a very, very big plus for all the potential combinations. We also now, recruited the team. We have a team in place. I joined the company 3 years ago. Very experienced people. Our head of CMC, for instance, Sharon, she is from the UK. She was a reviewer with the authorities for cell and gene therapy, so she has the experience from a regulatory perspective, but also from the production perspective. What, of course, is very important for a cell-based company. So we have a very clear strategy in place. We have been presenting the company to investors, but also potential partners, that basically are being exposed to the data, and now particularly with the new survival data that we're going to get in January, there's a lot of expectations and people waiting for that data. So I want to thank you, and I'm open for any questions.